Splatter EpiCheck real-time PCR using ABI 7500 FastDX instrument. Materials required but not provided. qPCR plates and adhesive film. qPCR plate holder, filtered tips, 1.5 milliliter tubes or strip tubes, mini centrifuge, qPCR plate spin down. Use the test form for reaction mix preparation. Calculate reaction mix for patient samples, 2 controls, and 1 extra for dead volume. Completely thaw the following reagents at room temperature: digested DNA samples, if they are frozen, PCR mixes A through H, Taq DNA polymerase. Cover qPCR tubes while thawing as they contain fluorescent dyes that are light sensitive. Once thawed, mix each tube before use. Do not vortex. Briefly spin down to remove drops from inside the lid. For PCR mixes preparation, you will need 8 clean tubes labeled A through H. Prepare assay mixes according to the table in the protocol and in the test form. Load qPCR mixes A through H according to the number of samples. Load Taq polymerase according to the number of samples. Mix each tube. Do not vortex. Mix digested DNA samples and controls as well. Briefly spin down to remove drops from inside the lid. Place a 96 well PCR plate on a stand. Make sure the top left corner well is A1. PCR mixes are loaded by rows. Pipette 15 microliters of assay mixes to the corresponding wells. Assay mix A into row A, assay mix B into row B, etc. The number of columns correspond to the number of patient samples and controls. In order to avoid cross-contamination, press the pipette plunger into its first stopping point only. Change tips between mixes to avoid cross-contamination. Perform visual inspection to ensure correct volumes in the plate. Use a full box of pipette tips to load digested samples and controls to allow visual tracking. Samples are loaded by columns. Pipette 10 microliters of each digested DNA sample and control to all wells of a column. 8 wells per sample. Cover the plate with optical adhesive film using plastic sealer. Make sure the plate is tightly sealed to avoid evaporation. Perform visual inspection of the volumes in the plate wells. Spin down the PCR plate. Insert the PCR plate into the instrument. Make sure to insert the plate in the right position so that top left well is A1.
Points to remember. Store digested DNA samples at -20 degrees Celsius for up to 30 days. In order to minimize cross-contaminations, separate areas, racks, and pipettes for template, non-template, and PCR mix setup. Each run analysis must include digested UC and NTC samples. Do not vortex reagents. Avoid exposure of PCR reagents to direct light and/or extreme temperatures for extended periods of time. It is recommended to use a full box, 96 count, of pipette tips to help with visual tracking when loading digested DNA samples and controls onto the PCR plate. Proceed to PCR instrument setup run analysis according to the instructions for use.